Lecture 6 will be on large intestine. Large intestine is the site where water is absorbed from indigestible residue of the liquid chyme, converting it into semi-solid stool or feces that is temporarily stored and allowed to accumulate until defecation occurs. And the large intestine is divided into three parts with regional specialization. So the large intestine is horseshoe shape, extends from the end of ileum to anus, lies inferior to the stomach and liver, and forms the frame of small intestine, and also called the large bowel, and it's about 1.5 meters long and 7.5 centimeter wide. So the function of large intestine is that it helps in water reabsorption. 2, it helps in compaction of intestinal content into feces. 3, it helps in absorption of important vitamins produced by bacteria, and also helps in storage of fecal material prior to defecation. So the part of the large intestine that are three are: 1, the cecum, which is a pouch-like first portion of the large intestine. We have the colon, which is the largest portion, and we have the rectum, which is the last part of the digestive tract, which is about 15 centimeter. So we now discuss the parts of the large intestine one by one. The cecum is the first part of the large intestine that is continuous with the ascending colon and is a blind expanded intestinal pouch. It's about 7.5 centimeter in length and breadth, and is located in the right lower quadrant inferior to the junction of terminal ileum and cecum, and they store materials and begins compaction when they receive material arriving from the ileum. Appendix, also called the falciform appendix, is a slender hollow appendage about 9 centimeter long, which is attached to the posterior medial surface of the cecum. And this mesoappendix is a mesentery that connects the appendix to the ileum and the cecum. And this mesentery are derived from the posterior side of terminal ileum, which attaches to cecum and proximal part of the appendix. So it's about 9 centimeter long, and it contains mass of lymphoid tissues. That is why appendix is also called a lymphoid organ. So this is the cecum, which is the first part of large intestine. I can see it continues superiorly with the ascending colon and is a blind intestinal pouch. And we have the cecum, which is found at the posterior aspect of the cecum, and also inferior to the ileocecal junction. And I said it's a mass of lymphoid tissue. That's why it's called a lymphoid organ. The colon is also a part of large intestine, and it has a larger diameter and thinner wall than small intestine. It has 4 parts, which are the ascending colon, transverse colon, descending colon, and sigmoid colon. The wall of the colon forms a series of pouches, also known as haustra, and these haustra permit expansion and elongation of colon. This colon has a muscles, and these muscles are 3 longitudinal bands of smooth muscles that run along the outer surfaces of the colon, deep to the serosa and similar to outer layer of the muscular intestinal. So these 3 longitudinal bands of smooth muscles are called the tinea coli. So the first part of the colon is the ascending colon, which begins at the superior border of the cecum. It passes superiorly on the right side of the abdominal cavity from the cecum to right lobe of liver, where it turns to the left at right colic flexure, also known as hepatic flexure. The ascending colon is located in the abdominal cavity and is covered by peritoneum anteriorly and on its side, and is approximately 2.5% of people has short mesentery. So this ascending colon has its own peritoneal covering anteriorly and on its side. There is no posterior covering by the peritoneum. The second part of the colon is the transverse colon, which is the longest and most mobile part of large intestine. It's approximately 45 centimeter long, and it crosses the abdomen from right colic flexure to the left colic flexure, where it bends inferiorly to become descending colon. The splenic flexure is usually more superior, more acute, and less mobile than hepatic flexure. This transverse colon lies anterior to inferior part of left kidney and attaches to diaphragm through phrenicocholic ligament. But the position of the transverse colon varies because it is movable. Being freely movable, it has a varying position. It's usually hanging to the level of umbilicus at the vertebral level 3, that is lumbar 3. But in tall people, the transverse colon may extend into pelvis. And this transverse colon is supported by transverse mesocolon and is separated from anterior abdominal wall by the greater omentum. The third part of the colon is the descending colon that occupies a secondarily peritoneal position between the splenic flexure and left iliac fossa, where it is continuous with the sigmoid colon. There is also a variation in its position too, because in approximately 30% of people, especially in the iliac fossa, descending colon has short mesentery, but not enough to cause volvulus of colon. And the descending colon is retroperitoneal, which is firmly attached to the abdominal wall. And the last part of the colon is the sigmoid colon, which is an S-shaped segment and is about 40 centimeter in length, about 15 to 40 centimeter in length. It starts at the sigmoid flexure, which link the descending colon and the rectum, and it extends from iliac fossa to sacral 3 segment, where it joins the rectum. It terminates at approximately 15 centimeter from anus, where it indicates rectosigmoid junction. The mesentery, it has a long mesentery and has considerable freedom of movement, especially at its middle part. It lies posterior to the urinary bladder, and as I said, the sigmoid colon empties into the rectum. So as we have discussed the large intestine, so this is the diagram showing the colon and the rectum. So this is the ostra, which is this wall, the pouches formed by the wall of the colon. So this is the ascending colon, which is the first part of the colon. And we have the transverse colon, which starts from which is one that will run through the transversely through the abdomen, and on the right side we have the hepatic flexure, and on the left side we have the splenic flexure, the cuff. And this transverse colon continues inferiorly with the descending colon. Descending colon, and descending colon moves continues inferiorly also and join with the sigmoid colon, which is S-shaped. And this sigmoid colon now join with the rectum. The rectum is a final and fixed terminal part of large intestine. It is about 15 centimeter and is an expandable organ for temporary storage of feces, which movement of fecal material into rectum triggers urge to defecate. The rectum continues proximally with the sigmoid colon and distal with the anal canal. That means the rectum is between the sigmoid colon and the anal canal. And the rectosigmoid junction is the inferior end of mesentery or sigmoid colon. So the rectum follows the curve of the sacrum and causes, which is the caudal vertebra, forming sacral flexure of rectum. But the part of the rectum is the part we call the ampulla of the rectum, which is the dilated terminal part of the rectum that receive and hold an accumulating fecal mass until it is expelled during defecation. The ability of the ampulla to relax to accommodate the initial and subsequent arrival of fecal material is another essential element of maintaining fecal continence. It is supported by levator ani and anacostia ligaments. There is a peritoneal covering around the rectum. The peritoneum covers the anterior and lateral surfaces of superior third, only the anterior surfaces of middle third, and no surfaces of inferior third. In male, the peritoneum of the rectum reflects to the posterior wall of urinary bladder, where it forms rectovesical pouch. And in female, the peritoneum reflects from rectum to posterior fornix of vagina, where it forms the rectouterine pouch. Anal canal is the last portion of the rectum, or the terminal part of large intestine and of the entire alimentary canal, and it contains small longitudinal fold called anal columns, and it's about 2.5 to 3.5 centimeter long. The anal canal is surrounded by external and internal anal sphincter, because we know that when the rectum that help to temporarily store the feces is full, this feces will move into the anal canal. So these two sphincter in the anal canal will control how the anal canal empty its contents. The first internal sphincter is the involuntary sphincter surrounding the superior two-thirds of the anal canal, which is found between the rectum and the anal canal, and its contraction is stimulated and maintained by sympathetic fibers, and its contraction inhibited by parasympathetic fiber. This sphincter contract most of the time to prevent leakage of fluid of flatus, and relaxes temporarily in response to distension of rectal ampulla, requiring voluntary contraction of external anal sphincter if defecation is not to occur and is sensitive to stretching. So this anal canal, the internal sphincter, is one that contracts when the rectum is full to allow the feces in the rectum to enter into the anal canal. But the external sphincter is a large voluntary sphincter that form a band on each side of the inferior two-thirds of the anal canal, so that when the feces is inside the anal canal, it is this sphincter that is voluntary in action, that an individual has a voluntary control over it, and it's one that decide that make the individual to decide the time to control the discharge of the content to the outside. So this external sphincter is sensitive to pain, touch, and temperature. This is the picture showing the rectum, and as we have discussed that anal canal is the terminal part of the large intestine, which is also the terminal part of the rectum. You can see this is the anal canal, which is the terminal part of that rectum, and the anal column is the small longitudinal folds of the anal canal. And at the anal canal, you can see the two sphincter, which is the internal anal sphincter and the external anal sphincter, and you can see the orifice, which is the anus. So that two sphincter are the one that control the control or regulate the way the discharge of content in the anal canal. So the internal anal sphincter is involuntary, while the external anal sphincter is voluntary. The anus is called the anal orifice, which is the exit of the anal canal, and it has a stratified squamous keratinized epithelium like epidermis of the skin. So function of large intestine in general, research of large intestine shows that there are less than 10% of nutrient absorption that occur in large intestine, and large intestine is the one that prepares the fecal material for ejection from the body. Other function of large intestine include is that it also plays roles in absorption. Those things absorbed in the large intestine are water reabsorption, bile salts reabsorption in the cecum and transported in blood to the liver, absorption of vitamins produced by bacteria, and absorption of organic waste. Vitamins are organic molecules and they are important cofactors of coenzyme metabolism. So normal bacteria in colon make three vitamins that supplement the diet. The vitamins produced in the large intestine are vitamin K, which is fat soluble and required by liver for synthesizing four clotting factors including prothrombin. Another vitamin produced by the liver by the large intestine is biotin, which is water soluble and is important in glucose metabolism. And the third vitamin produced in the large intestine is pantothenic acid, which is B5, also water soluble, and this vitamins is required in manufacture of steroid hormones and some neurotransmitters. So how does large intestine absorb organic waste? Bacteria convert bilirubin, which is the broken down of uh, dead or old red blood cell, the product from the red blood cell broken down. These bilirubin are converted to urobilinogens and stachobilinogens. So these urobilinogens absorbed into the bloodstream are excreted in urine. Urobilinogens and stachobilinogens in colon convert to urobilins and stachobilins when they are exposed to oxygen. The role of large intestine in toxins. Bacteria also that are found in large intestine break down peptides in feces and they generate the following compound. We have the ammonia as soluble ammonium ions, indole and scatol, which are nitrogenous compounds responsible for odor of feces, and also hydrogen sulfide, gases that produce rotten egg odor. Bacteria also feed on indigestible carbohydrates, which are complex polysaccharide and produce flatus or intestinal gas in large intestine. So the large intestine, instead of this toxin deposited in our body, they help us to find a way to break them down and release them out of the body. Movement of the large intestine. We also have two reflexes, which are gastroileal and gastroenteric reflexes that move materials into cecum while we eat. So movement from cecum to transverse colon is very slow, allowing hours for water absorption. Peristaltic waves move material along the length of the colon, and segmentation movement, which is the ostral churning, means the content of the adjacent ostra. Movement from the transverse colon through the rest of large intestine results from powerful peristaltic contraction, which is a mass movement. And stimulus is distension of stomach and duodenum relay over intestinal nerve plexus. Distension of the rectal wall triggers defecation reflex, and there are two positive feedback loops. Both loop triggers by stretch receptor in rectum, in which one stretch it will release the content to the anal canal for defecation reflex to take place. Elimination of feces. This one requires relaxation of internal and external anal sphincter. Reflexes open internal sphincter and close external sphincter. but opening of external sphincter requires conscious effort, which is a voluntary action. Lecture 7 is on accessory organs of digestion. And these accessory organs of digestion are the pancreas, liver, and gallbladder that assist with the digestive process in the small intestine. So the first one we'll be discussing is the pancreas. And the pancreas is the one that lies posterior to the stomach and is bound to the posterior wall of the abdominal cavity, wrapped in thin connective tissue capsule. It's an elongated accessory digestive gland, and it lies retroperitoneally and transversely across the posterior abdominal wall, posterior to stomach, between the duodenum on the right and spleen on the left. The pancreas has two structures. We have the pancreatic acini and pancreatic islet. The pancreatic acini are blind pockets that are lined with simple cuboidal epithelium, and they contain the scattered pancreatic islet. While the pancreatic islet is the endocrine tissue of pancreas and that produce the hormones from the pancreas. This is the picture showing the pancreas fitted in the C-shape of the duodenum, and we are going to use this figure to explain the parts and the ducts of the pancreas. The first part of the pancreas we are going to mention is the head of the pancreas, which is the expanded part of the gland that is embraced by C-shape of curve of the duodenum, and it firmly attaches to the duodenum. The second part we are going to mention is the body of the pancreas, which lies to the left of spermatocytic vessels, and the posterior surface of the body of the pancreas is devoid of peritoneum and is in contact with the aorta. The anterior surface of the body is covered by peritoneum and lies in floor of omental bursa and form part of the stomach bed. And the last part is the tail of the pancreas, which lies anterior to the left kidney, where it is closely related to the splenic ileum and left colic flexion. Tail of the pancreas is relatively mobile. So the duct, there are two ducts along the pancreas. We have the main pancreatic duct and the accessory pancreatic duct. The main pancreatic duct begins in the tail of the pancreas and runs through the gland to the head, turns inferiorly and closely related to the bile duct. The main pancreatic duct and the bile duct most of the time unite to form short hepatopancreatic ampulla, which opens into the duodenum. The second one is the accessory pancreatic duct, which opens into the duodenum and carries pancreatic juice, mostly in some people where the main pancreatic duct are smaller and the two are not connected. So the pancreatic secretion. Pancreas produce two types of secretion, and this secretion, which is called the pancreatic juice, is about 1,000 milliliter per day. The two secretions are the endocrine secretion, which contain the pancreatic enzymes, and this pancreatic juice are from acinal cells that enters the duodenum through main and accessory pancreatic duct, while the endocrine secretion, which are the glucagon and insulin, come from the pancreatic islet that enters into the blood. The pancreatic enzymes are the pancreatic alpha amylase, which is a carbohydrate enzyme that break down starches, and its role is similar to salivary amylase from the salivary gland. Second one is the pancreatic lipase that break down the complex lipid and release products, which is fatty acid, that are easily absorbed. The third one is the nucleases that break down nucleic acid, and the fourth one is the proteolytic enzymes that break certain protein apart. Proteases break large protein complexes. Peptidases break small peptides into amino acid. 70% of all pancreatic enzyme production secreted as inactive proenzyme and activated after reaching the small intestine. The second accessory organ that will be discussed is the liver. And liver is the largest visceral organ after the skin, which is the largest single organ. And it weighs approximately 1.5 kilogram and accounts for approximately 2.5% of adults' body weight. In late fetus, in which it serves as hemopoietic organ, it is proportionately twice as large, 5% of body weight. Liver is located from early childhood onward. It occupies all the right hypochondriac and epigastrium, and it extends into left hypochondrium inferior to diaphragm, which separates it from pleura, lungs, pericardium, and the heart. When you're looking at the anatomy of the liver is wrapped in tough fibrous capsule and is covered by viscera, peritoneum, and also divided into lobes. So this is a diagram showing the liver. We have the two lobes of the liver, the right and the left lobe. We have the surfaces we are going to discuss. So now let us look. This is the anterior surface of the liver. There are two surfaces, which are the diaphragmatic and the visceral surface. So the one we are seeing now is the diaphragmatic surface, which is the smooth and dome shape and is related to the concavity of the inferior surface of the diaphragm. This surface is covered with viscera, peritoneum, except in the bare area of liver, where it lies in direct contact with the diaphragm. So the liver also has lobes at this anterior surface. It is two lobes externally, which is the right and the left lobes. But the posterior surface, also known as the visceral surface, this visceral surface is covered with peritoneum, except at the fossa for gallbladder and portal hepatis, which is a transverse feature where vessels, hepatic nerve plexus, and hepatic duct that supply and drain the liver enter and leave it. The visceral surfaces bear multiple features and impression from contact with other organs. That is why there are some impressions that can be seen on these posterior surfaces of the liver, which shows the contact it has with other organs at these visceral surfaces. This posterior surfaces, which is the visceral surfaces, has their own lobes. You can see there are 4 lobes that can be seen around this area. We have the major right and left lobe, and there are 2 accessory lobes, which is due to because there are 2 features there. We have the right and left sagittal features that surround the transverse portal hepatis that demarcate the 2 accessory lobes. We have the first one, which is the quadrate lobe, that is located anteriorly and inferiorly, And we have the caudate lobe that is located superiorly and posteriorly. The liver has their own blood supply. We have the hepatic blood supply, which is the 1/3 of the blood supply, and the 2/3 is from the hepatic portal vein. So the hepatic artery is the one that accounts for 20 to 25% of blood received by liver, and is from the hepatic artery proper. While the 75 to 80% of the liver is from the hepatic portal vein that originates from the esophagus, stomach, small intestine, and the large intestine. This hepatic portal vein contains 40% more oxygen than blood returning to heart, which help to sustain the liver. And the liver is drained by the hepatic vein. The histological organization of the liver shows that there are lobules in the liver, which is the basic unit of the liver. Each lobe is divided by connective tissue into about 100,000 liver lobules, which is about 1 millimeter diameter each. And they are hexagonal in cross section and they have 6 portal areas called hepatic triads, one at each corner of the lobule. So the portal area of the liver contains 3 structures. You have the branch of hepatic portal vein, branch of hepatic artery proper, and small branch of the bile duct that forms the triad, which is the portal area. The physiology of the liver, or the function of the liver, are categorized into 3. They help in metabolic regulation, hematological regulation, and bile production. And this function will be spread across all the vital functions of the liver that will be listed. So these are the functions of the liver. The digestive and metabolic functions of the liver are: 1. It helps in synthesis and secretion of bile. It helps to store the glycogen and lipid reserve. It helps to maintain the normal blood level of glucose, amino acid, and fatty acid. Helps in synthesis and interconversion of nutrient types. Helps in transamination of amino acid or conversion of carbohydrate to lipids. Helps in synthesis and release of cholesterol bound to transport protein. It helps to inactivate toxins, serve as iron reserve storage, and also serve as storage for fat soluble vitamins. The other major functions liver perform is that it helps to synthesize plasma protein, the clotting factors, helps to synthesize the inactive hormone angiotensinogen, also helps to phagocytose the damaged red blood cell by Kupffer cells, helps in blood storage, absorption and breakdown of circulating hormones and immunoglobulins, and also absorption and inactivation of lipid soluble drugs. The next accessory organs of digestion is the gallbladder, which is a pear-shaped muscular sac, and this gallbladder is about 7 to 10 centimeter long, that lies in the fossa for gallbladder and facial surface of the liver, and this shallow fossa lies at the junction of right and left part of the liver. It is pear-shaped, as I said, and it can hold up to 50 milliliter of bile. And so using the diagram, this is the diagram showing the gallbladder. This gallbladder has 3 parts. We have the fundus, the body, and the neck. The fundus is the wide end of organ that projects from the inferior border of the liver and is usually located at the top of the right ninth costal cartilage. The body is one that contacts the facial surface of the liver, transverse colon, and superior part of the duodenum. And neck is the one that is narrowed and tapered, directed towards the portal hepatis, and make an S shape and joins the cystic dot. So that cystic dot is one that is shown in this figure, which is about 3 to 4 centimeters long, that connects the neck of the gallbladder to the common hepatic dot. So the function of the gallbladder is that it helps in bile storage, releases the bile into the duodenum, but only under stimulation of hormone cholecystokinin, as mentioned in the hormone of the digestive tract. This uh, gallbladder also plays a role in which this cholecystokinin plays a role in such a way that it's when hepatopancreatic sphincter remain closed, so that bile exiting the liver in common hepatic duct cannot flow through the common bile duct into the duodenum. But with the help of this cholecystokinin, it opens this sphincter to allow the bile to be released into the duodenum. So when this sphincter is closed, the bile enter the cystic duct and is stored in the gallbladder. The physiology of the bladder is that it contains about 40 to 70 milliliter bile, and the bile composition uh, gradually changes in gallbladder. Water is absorbed and bile salt and solute become concentrated. So the component of bile is that every 500 milliliter of bile are secreted by liver daily, and they contain water, mineral salts, mucus, bile pigments, mainly bilirubin, bile salt, and cholesterol.